Hi, I'm Hugh Patterton. I'm the director of the Center for Bioinformatics and Computational Biology here at Stellenbosch University, and it's really a pleasure to tell you more about the exciting new field of bioinformatics and computational biology. So the first question you probably want to ask is, well, what is bioinformatics? So to basically phrase that quite succinctly is that bioinformatics is an interdisciplinary science. So interdisciplinary means that it's made up, or that you require skills from a number of other fundamental sciences, including mathematics, stats, computer science, biochemistry, genetics, etc. So it's an interdisciplinary science, and it's a science that has to do with the gathering, manipulation, storing, retrieving, and very importantly, the analysis of biological data using computers. So you would all be very aware that we're living in the era of big data. And in fact, if you look at the rate at which we doing Google searches, which we're sending tweets on Twitter, the rate at which we're reading messages on Facebook, the pace at which we are reading and downloading pages from Wikipedia, the rate at which our cell phones are transmitting location data that is used to calculate traffic density and so on. If you add all of that up, then you get to a sum total that in the last year, we've generated about 10 to the power 18, that's 1 quintillion bytes of data every minute. So essentially 10 to the power of 18 is equivalent to a million notebook computers. So last year, every 1 minute, we filled up the hard disks of a million notebook computers with all the data that we're generating. So an important part of big data is basically biological big data. And in fact, bioinformatics can be seen as the data science that's applied to biological data. So with biological data in this slide, basically I'm showing you the rate at which we're generating new DNA sequence data. So you can see that if you look at the yellow trace, that in approximately 2008, there's suddenly a dramatic increase in the rate in which we're producing DNA sequence data. And that is because at that point, we basically made a change from a slow old type of technology to a multiplex technology to generate DNA sequences. We basically parallelized the technology, generating DNA sequences from millions of DNA molecules simultaneously, millions of different DNA molecules simultaneously. And in fact, the amount of data, DNA sequence data that we generated is equivalent, again, to 1 exabyte of data. So over this slide, I'm indicating it as an exabase pair. A base pair and a byte is not entirely equivalent. 1 base pair encodes approximately 2 bytes of data. But if you see it sort of roughly, order of magnitude, we've generated about an exabyte of DNA sequence data in 1 year. So you may want to ask yourself, what is this information useful for? Why do we need this genome sequence data? So the first thing that you will basically encounter when you look at sequence data and you try and make sense of it is that the sequence data is exactly the type of data that I'm showing you on this slide. So it's essentially a sequence of GATs and Cs. And in the English language, you can think of a meaning as different letters being collected together in words. And when we read these specific orders of letters in a word, that makes sense to us. And by basically using these words, we can communicate information from one person to another. So that is using English language or Afrikaans language or any other language for that matter, doesn't really matter. Here, the language we're looking at on this slide is basically genome language, which is composed of the 4 letters G, A, T, and C. And the point is, looking at this language, We don't see any punctuation. It's not clear where the words start, where the words end. We cannot see any paragraphs. So we need to make use of a computational approach to look at this collection of letters and make sense of it. Where do the words start? Where do the words end? You can think of the words as the equivalent of genes, where genes encode a single protein that can either be structural protein or that can be an enzyme that catalyzes a specific chemical reaction. So an important point with looking at genome sequencing data is how do we interpret that data? How do we actually get to understand what that data means? And that is where bioinformatics come in, where you basically use a bioinformatics approach to analyze the GATNC sequence and basically identify the start positions of genes, the end positions of genes, and other regulatory sequences that control the rate at which genes are expressed under specific sets of conditions. So once you've applied this type of bioinformatics approach to a genome sequence, you end up with what is known as an annotated genome. So an annotated genome is simply the genome sequence, and we know where all the genes are, and we know where they start and end, and we know where additional control elements in the genome are. So on this slide, I'm showing you what is known as a genome browser, which is basically a type of software that runs within a web page that allows you to look at a genome. And this particular genome we're looking at here is in fact a little part of chromosome 3 of Baker's yeast, Saccharomyces cerevisiae, which is a favorite genetic model organism. And the red blocks that you see are the location of the individual genes, and the arrows that are linked to the genes indicate the direction in which these genes are basically transcribed. So you'll know that DNA has two strands, a Watson strand and a Crick strand, and any of those two strands can encode a gene, which is the reason why sometimes it goes in one direction and at other times another gene will actually go in the other direction. So this is an annotated genome, and although that is quite interesting and one can basically look at that and learn a lot and do an analysis perhaps of the regulation of specific genes and co-regulation of genes, etc. An important aspect of genomes also includes not the genome of a single individual, but if you actually sequence the genomes of a number of individuals in a group. So over here, I'm showing you just a teeny weeny part of a genome. In this case, it's from C. elegans, which is a nematode. And what this slide shows is basically the teeny bit of the genome of the organism that has been aligned between the different individuals. So the genomes of different individuals of this organism has been sequenced, and these genomes have now been aligned. And if you align them, which means that all the corresponding nucleotides are basically aligned with the identical corresponding nucleotides in the other genomes, and you run a specific set of software on the aligned genomes, then you find that there are some positions where some of the individuals actually differ from that of the population. So, for instance, in this case, you can see, looking at the aligned sequences at the bottom, there are a couple of positions where there are lone T's and A's and C's, etc., in those gray bars. And those are positions in the individual genomes where the nucleotide sequence of that particular individual differs from that of the population. So these individual mutations are called single nucleotide polymorphisms, or SNPs. And it is possible to look at these individual SNPs or patterns of SNPs and the groups within which they've been identified, and see if there's any correspondence between these SNPs and, for instance, a diseased state. And this type of study is known as a Genomewide association study, where you associate a diseased state with a specific SNP. And in fact, we have identified many, many SNPs that are associated with a multitude of human diseases. And here I'm just showing you a very, very abbreviated list of some diseases associated with specific SNPs. So for instance, if you run down the list, you'll see that for type 2 diabetes, there's actually a SNP that occurs in the gene KCNQ1, and in the right-handmost column, the RS number is basically the reference SNP number, and a reference SNP has been assigned to this specific disease. And if you look at some of the other rows, there are multiple other diseases that I'm also showing where the disease state has been associated with a specific SNP, including bipolar disorder, coronary heart disease, schizophrenia. So there are multiple diseases, and again, I emphasize this is but a tiny abbreviated list of human diseases that have been mapped to specific mutations or SNPs in our genomes. So that immediately begs the question, what about sequencing new natal genomes? Would that hold any type of advantage? So for instance, directly after baby's birth, it gets taken through a series of tests before it gets passed on down the line of authority. Perhaps one should consider one of the things that one can do is just take a little bit of a swab from the inside of the baby's mouth and basically use that to sequence the baby's genome. And this has in fact been done. So there were a couple of experimental trials in the US, and one of these trials were called BabySeq. And by looking at the baby genome, it was quite astonishing that 3.5% of the healthy babies had adult onset conditions. So some of these adult onset conditions, so these are only diseases that will manifest themselves in adulthood. So some of these diseases include hereditary breast cancer, ovarian cancer, and Lynch syndrome. And quite strikingly, 88% of the 116 newborns that formed part of the study were actually carriers of recessive conditions, which means if they would happen to choose a life partner and have a baby, then there is a risk, depending on the genomic composition of that partner, there may be a risk associated with that condition being present in the baby. Additionally, 5% of the babies had pharmacogenetic variants that could influence the drug dosing decisions. So different genes encode different proteins, and some of these proteins are enzymes that are involved in metabolic pathways, and some of these metabolic pathways have to do with the metabolism of compounds such as drugs. And some people are more efficient at metabolizing drugs, and other people are less efficient at metabolizing drugs. So often you have that drugs that are administered will have different effects or efficacies on different patients. And you can trace this back to genome sequences, and these babies showed that 5% had pharmacogenetic variants that could influence that drug dosing decisions. 6 of the newborns had variants that were related to hypertrophic cardiomyopathies. 4 newborns had variants in muscle protein, and you can read the rest of the text there. Basically, the point that the slide makes is that in this very tiny group, there were a multitude of diseases that were identified associated with a sizable proportion of the newborns that would only exhibit itself in adulthood, and some of these diseases were quite serious. So it just seems that it would make a lot of sense to actually sequence the genomes of newborn babies directly after birth and analyze the genomes and analyze the disease states to which the babies have a higher risk, because then you can make lifestyle changes. You can basically organize early intervention, etc., etc. So once you know that there's an increased risk for the development of a specific state, then you can manage that state much better. So it has tremendous value to actually determine the genome sequences of newborn babies for health reasons. So this is simply the Afrikaans translation of that rather wordy slide, for those of you who prefer to read the Afrikaans. The other point is that if you look at different groups, then different groupings have often different patterns of SNPs. So often when we look at risk profiles, we look at the risk profile of the typical white male. But it's also true that if you look at females or if you look at other population groups, then the SNP profiles differ, and also the risks associated with specific profiles differ. So for this reason, it is imperative that we actually generate enough sequence data from diverse human population groups and analyze the SNP patterns and the disease states that are associated with those SNP patterns, because there is very, very clearly a difference between the population groups and the patterns and mapping to specific disease states in these groups. So we've actually gone quite a way to do this. One of the projects that we've completed, which is known as the 1,000 Genomes Project, where 1,000 human genomes essentially were sequenced, gave a tremendous amount of information on the presence of specific SNPs within different population groups at different geographic locations on our planet. And we've actually gone way beyond this. So we've generated far more than 1,000 human genomes at this point. We're probably running into tens of thousands of human genomes distributed all over the planet, and of various groups that may exhibit resistance to specific disease states, to get a more complete map of different SNP patterns to specific human diseases. So this is an important aspect, and basically to do this type of study, where you take genome sequences and you see if there's any association, statistically significant association with specific diseases, is a specialized field within the discipline of bioinformatics. So another important point is that any two individuals differ by at least 20,000 SNPs. And these different SNPs can have an impact on your response towards specific medication. So some individuals with specific SNP patterns would respond well to medicine A, and other individuals may not respond well to medicine A, but respond well to medicine B. And it's becoming increasingly important to actually know the pharmacogenomic profile of an individual, to know what type of medicine will work more efficiently in that individual. So this is also the area that's known as personalized medicine. So in summary, what do bioinformaticians do? So we manage the storage of biological data in appropriate formats. We design databases and interfaces that allow the very efficient retrieval of data from those databases. We develop algorithms which thinking of better ways, for instance, to very quickly analyze where genes start. So once we've developed that algorithm, then that algorithm can, for instance, be applied in a new analysis method or a new software tool that we can code, or we can use established software tools to analyze biological data sets. We also do research in cutting-edge methods such as machine learning and artificial intelligence, and apply that to biological problems. So some of you may be aware that towards the end of last year, a research group within Alphabet, which is the parent company of Google, developed a program that's known as AlphaFold 2, that's based on machine learning, and essentially that takes the primary sequence of amino acids that form a protein, and from that sequence of amino acids, it predicts by looking at the interactions and energies of potential interactions and comparing that to very large data sets that was used to train the machine learning program. It uses that to predict in what shape the protein could fold. So that is an age-old and really very, very difficult problem in biology, to basically take the amino acid sequence of a protein and then derive from that information the final 3D structure of the protein. So that has tremendous application and tremendous value in biology to understand that. And you can also imagine it's possible to work in the reverse. So for instance, you look at a specific chemical reaction, you can think of a protein shape that will allow a specific spatial distribution of charges that will catalyze the chemical reaction. And then once you've designed that protein with the distribution of charges at specific locations, you can then work back and see, well, what is the sequence of the protein that will actually form that catalyst. So it's a really exciting era also in biotechnology that we're entering, where we can actually design protein sequences de novo, or from the start, to basically fold in specific shapes to catalyze specific reactions. And some of those reactions may be reactions that do not necessarily or normally occur within living cells. It may be quite novel types of chemical reactions that we may have an interest in introducing. The final point, I think, that in terms of the summary of bioinformatics is that in the end, given all these activities, what bioinformatics is really about is about asking novel and interesting questions about living systems, and to think of ways to answer these that contribute to human knowledge. So it's basically using computers to understand living systems. And at this point in the slides, I've emphasized genomes, genome sequences, SNPs, and the relationship between SNPs and human health quite extensively. You should be aware that that is a tiny part of the field of bioinformatics. Bioinformatics are equally applicable to protein sequences, to structures, to protein structures, to metabolism, to gene expression. So there are lots of other specialized fields looking at different types of molecules and different biological processes to which bioinformatics is applicable. So at Stellenbosch University, we're offering quite a comprehensive pipeline to train as a bioinformatician. We're starting off with an interdisciplinary BSc. So the interdisciplinary BSc will basically give you training in your first year in computer science, in maths, probability and statistics, and chemistry, biology. In the second year, you'll be exposed to statistics, to biochemistry, to genetics, computer science, and in your third year to computer science, biochemistry, and genetics. And if it's an area that really grabs you, you can continue your training in the field by doing a BSc Honours in Bioinformatics and Computational Biology, and you can follow that up with a master's degree in Bioinformatics and Computational Biology, and you can basically proceed to train up to a doctoral level with a PhD in Bioinformatics and Computational Biology, all at Stellenbosch University. So in order to study, to register for the interdisciplinary BSc in Bioinformatics and Computational Biology, you're going to need a National Senior Certificate or equivalent with university exemption at an average matric mark of 65%. And you're going to need 70% for your matric maths, and 50% for physical science, and 50% for English or Afrikaans home language or first additional language. The valid question is, after you've studied bioinformatics and computational biology, where do you work? Where would you actually get a job? So there are a number of possibilities. I think the two big employers would be academia and then health and biotechnology industries. So in academia, you can be either an academic with your own research group doing fundamental research in bioinformatics, or you can be a member of an established research group, and that research group may be generating a lot of biological data and they require your assistance as a bioinformatician to analyze and help them analyze their data. Or you can work in a central core facility at university, which is maybe associated with a sequencing facility or mass spectrometry facility, where you are involved in helping people analyze the data, testing specifically hypotheses against the data. There are oodles of opportunities in private enterprise and health and biotechnology industry all over the world. Some of the activities would include making a bioinformatics consultancy company to either help academic institutes or private enterprise with solving bioinformatics problems. You can be involved in bioinformatics software development. We're actually coding or writing programs, and that can be either in your own company or as part of a larger company. A company such as Illumina, for instance, that is involved in the manufacture of technology that's used in DNA sequencing would employ many bioinformaticians to help them develop software that they use to control the instruments and analyze the data generated by those instruments. You can be involved in drug design in big pharmacological industry. So an important aspect of drug design is you know what the protein looks like that you want to influence, and that protein may bind the substrate, and you're looking for an alternative substrate that would interfere with the ability of that protein to bind the substrate, or that would somehow modify the protein shape so that it cannot catalyze a specific reaction anymore. And a lot of the work in looking at possible small molecules that can perform this task involve bioinformatics, where you basically sit in front of a computer and you model the protein and you model the small molecule and you look at the interaction and binding, which is known as docking. So that's an important aspect in drug design. You can be involved in genome analysis. So increasingly, it's going to become something that is mainstream, that humans will have their genomes sequenced and analyzed, and the bioinformatician would be involved in that analysis and perhaps also communicating with medical practitioner. Additional area would be in biomarker discovery, which would be the identification of specific proteins. Pharmacogenomics, which has to do with the susceptibility of specific individuals with different genomes to different pharmacological agents, and that includes the area of personalized medicine, where different people respond differently to different treatments. So that is a short list of areas where bioinformaticians do find work all over sort of multinational companies. If this is an area that you thought was or is quite interesting and you want additional information, you're very welcome to contact me. Once again, my name is Hugh Patterton, and I'm the director of the Center for Bioinformatics and Computational Biology. My email address is hpatterton@sun.ac.za. You're very welcome to drop me an email or give me a call, or you're welcome to drop by. I'm in the Peril building on the Stellenbosch University campus, and it will be a pleasure to sit down and discuss the exciting new field of bioinformatics and computational biology with you. So thanks very much for your attention, and I'm looking forward to actually see some of you in the first year class for bioinformatics this year and in following years. Thank you very much.